Today we're going to start learning about dihybrid crosses, and we're going to tie that in with our Mendelian corn lab. In this illustration you will see what we'll be working with in our corn lab today, in which case we have a parental generation here at the top. Remember that the parental generation is always going to be homozygous. In this case they are crossing one parent who has dark seeds that are also smooth. And on the other side, we can see this yellow corn that's wrinkled. This is going to be slightly different from the parental generation we'll use in our lab today. If you look down here at the note, you'll see that we use dark wrinkled seeds and yellow smooth seeds for our parental generation. However, it doesn't really matter which one we choose because the F1 generation is going to always look the same, because the F1 generation is always heterozygous. And since we're dealing with complete dominance in our assignment today, you will see that no matter what happened, you always came out with these purple smooth seeds. Now, when I cross the F1 generation together, I cross the heterozygotes together, I am always going to get the F2 generation, and that F2 generation is always going to be a mixture of all possible combinations. And so when I look down here, I can see yellow smooth, I can see dark wrinkled, we have yellow wrinkled, and we have dark smooth. So I get all four combinations possible in this particular cross by the time I get to the F2 generation. But we want to take a step back and we want to talk about how we would go through and work out this information using a Punnett square. One thing to always remember, and you'll see this on a number of our slides, is that the gametes, which go on the outside, are going to have 2 letters now, and that the zygotes, which we would find on the inside, because they are the potential offspring, are going to have 4 letters. So everything I draw on the outside, 2 letters, everything on the inside, 4 letters. And while we normally wouldn't do a Punnett square to show the parental cross going to the F1 generation, we're going to start at the beginning and go through that also. Now, one thing you're always going to want to make sure you have is a key. And in today's lab, you will be coming up with a key also. You will have the trait that you're going to be looking at. You're going to have the phenotype, and you're going to have the allele, or the letter that you are going to use. Now today we're going to choose 2 different traits. We're going to say we're looking at an animal, and they have a difference in coat color, as well as in the height that that animal can grow. So those would be our 2 traits. Sometimes we refer to those as our, that's what our genes are coding for. We have one gene coding for coat color, we have one coding for height. Then I have choices within there. These are my phenotypes. Remember, the phenotypes are always written out as words. And so for this particular animal, I'm going to have 2 different choices. I could have an animal with a dark coat or a white coat. And I can have 2 different heights. This animal can either be short or tall. I then have to come up with the alleles, those letters that I want to use. Now, coat color is going to be using the same letter. One will be a capital though, one will be a lowercase. Same thing for height. Height will have the same letter, but you'll have a capital letter or a lowercase letter depending on which one is dominant and which one is recessive. And when I look here, this first one, I chose D to represent dark because that is our dominant phenotype. And that's what happens a lot of times when you look at these letter combinations. They really go with the dominant phenotype. W, I wouldn't use a W for white because I'm dealing with coat color. And so I'm going to use a little d there. However, when I look at height, I don't want to actually use an S because an S is really hard to tell the difference between a capital and a lowercase letter. And so I just randomly picked another letter to use, which is perfectly fine because I said in my key what I'm going to be using. And in this case, I used a capital M and a lowercase m. Big M for short, little m for tall. So here I want to talk about bad letter combinations. In this experiment, you are either going to be printing out the lab and writing in everything by hand, or you could be typing in everything. In either case, you're going to be uploading this to Moodle and expecting me, the instructor, to be able to tell the difference between your dominant and your recessive alleles. But if you choose bad letter combinations, like big C, big S, little s, and so forth, it can be really difficult to tell, looking at those letters, which is dominant and which is recessive. And so I would like you to choose letters that look differently as a capital and lowercase, like the big D, the big M, little m. And this will ensure that I can interpret what you are telling me in your lab properly. We want to talk about the two different ways our parental genotypes and phenotypes can look and still be homozygous. The first option is to have the phenotype for one parent be dark and short. If I look at my table, I can see that those are both dominant phenotypes. And so the dark to be a parental genotype has to be big D, as well as big M, big M. The second parent could be white and tall. To be homozygous and white, you would need to have both the little d and another little d. It's the only way that the recessive can show up in the phenotype. The same thing happens for tall. I have to have both little m, little m. I could have two different types of parental phenotypes and the resulting genotypes and still get the same result by the end of the experiment. And so my other option would have been a dark, tall individual. This is still homozygous because the big D, that's homozygous. The little m is homozygous. You have to remember these are separate. And for this experiment that we're doing, as well as for this example, we are assuming that they are on separate chromosomes, and the results do support that. And they're going to be independently sorted from one another. If you remember from the discussion of meiosis, in metaphase 1, in meiosis 1, these chromosomes lined up 2 by 2 by 2 in their X or Y form. And you had a 50% chance that the chromosome that that individual had gotten from its dad lined up on the right or lined up on the left. And every single pair of chromosomes has that 50-50 chance. And so we can have multiple options. But we are looking at this going tall and dark are homozygous. White and short would also be homozygous. Little d, big M, big M. The next thing we'll be talking about is how we come up with the gametes for the Punnett squares. Remember, we're going through the process of meiosis, and so we're going to go from 4 letters in the parent down to 2 letters for the egg and sperm cells, those gametes that go on the outside of the box. When I look at the parental generation, I can see that the only letter parent number 1 can give is a big D for the dark coat color, and it can only give a big M for the short height. In addition, the other parent can only give a little d for the color white, and it can only give a little m for the tall height. Because of that, when I flip to this slide, you will see what the outside of the boxes are going to look like. Remember, the parental gametes, 2 letters, and I should have both a D and an M there in my example. And yet, the zygotes that go inside with the 4 letters should have 2 Ds and 2 Ms, one from each side. Here I am filling in the Punnett square. So across the top, I want to drag down that big D and the big M in that first column. And when I look at all the boxes underneath, I will see those two letters in the resulting squares. I then go off to the left side, and you can see I want to drag across that little d and that little m into the resulting boxes. And for this particular example, I color-coded both the parents. So I did one in black and I did the other one in red coloring. Now, since this is the F1 generation inside the box, you're going to see that the genotype is all the same. They were all heterozygous, big D, big M, little m. And because each of these had a capital letter, a big D and a big M, that means their phenotype is all going to be the same. They're all going to code for dark, short individuals. So now I want to take that F1 generation and I want to cross them in order to find the F2 generation within the box. And so how do I find the gamete combination now? It's not as simple as when I cross the parental generation. So here's how I like to do it. I take the 4 letters for that particular individual, big D, big M, little m, and I lay it out this way. And then underneath it, I start to draw lines. I can only use each letter twice. And so here I drew black lines under a big D and a big M, giving me big D, big M. And when I do this, remember, I have to have one of each letter. I can't have 2 D's or 2 M's as one of my gametes, in which case something went horribly wrong during meiosis, and I'm not going to get the correct answers for this Punnett square. So I have my first set of gametes, and the next thing I'm going to do is still use that big D, and I'm going to draw another line under it, and then I'm going to move on to the other M, that little m, and I drew those in red. So big D, little m is my next set of gametes. Now, since the big D had 2 lines under it, I had to move over. I had to go to the little d. And so here you can see I drew green lines under the little d and the big M, giving me my third set of gametes. And now I can't use that big M anymore. It has 2 lines. And so I continue along, and I can still use the little d and the little m. I drew those in purple, and that is going to be my 4th letter combination. And so here you can see that I put those across the top and down the side. Since both parents were of the F1 generation and they were both heterozygous, it's going to be the same across as well as down the left side. This is standard. This is how it'll be set up in your textbook, how it was set up in the PowerPoint we went over for Mendelian genetics, and is typically considered the standard. If you put things out of order though for your particular lab, that is okay. I know what the results should be and I can interpret them however you fill these out. Now here we want to see the completed chart. And so you can see once again I dragged that big D, big M down, that first column. And then I dragged that big D, big M across that first row. And then I keep doing that until every single box is filled in with 4 letters. Now if you look at where I put the orange arrows, you can see that I dragged that little d, big M all the way down to the box that the arrow ends in. And then I dragged that little d, little m all the way over to finish filling in the box. When I did that, I can then interpret the phenotype because these have little d's that corresponded to the white coat color, and because there's one big M that corresponded to the short height. Now here is the final results when I filled in the entire chart, and I color-coded it this time in order to show you what the different phenotypes are going to be. and then I put the results off on the side. When I look here, everything that I coded in yellow is going to be dark and short. For example, they all have at least one capital letter D and one capital letter M in them. Therefore, they're going to be dark and short because you only need one capital letter in order to show that phenotype. There are 3 that are going to be dark and tall. Those are the ones in orange. So you can see they have at least one capital letter D and both of the M's are lowercase. The 3 that are white and short that are in green on the chart, you can see I have 2 lowercase d's and at least one capital M. And then you have the oddball. You have the one that is homozygous recessive for both traits, which means it is little d, little m, 1/16th chance of this happening, and that is the blue square at the very bottom. We'll now move into talking about our lab, the Mendelian Corn Lab, looking at dihybrid crosses in corn. You can see from this corn cob that we are going to have 4 different phenotypes. We're going to have yellow smooth, yellow wrinkled, dark smooth, and dark wrinkled. During this lab, you will see that I tried to underline the important parts for what you need to accomplish. You'll see the first thing you're going to be doing on page 1 is writing in the phenotypes in the appropriate boxes that you see at the bottom half of this slide. When we go to the word problem, you will see underlined that there are 2 true breeding plants. Remember, true breeding is the same as pure breeding or homozygous. Because it's homozygous, these must be the parents or parental generation. One of them was yellow and smooth, and this was crossed with another true breeding plant, which was dark and wrinkled. Remember, the cross signified by the X that you see on the diagram means that they were bred together and produced offspring. Now, all the offspring, as you continue reading, all the seeds produced were dark and smooth. And then when these F1 seeds were planted, you got the ear of corn that you see on the screen in the illustrations that you will be counting. And all of those variations need to go into the box labeled F2. Now, as we continue, we're going to see the second half of page 1. Here, you're going to have to answer question number 2, which states that based on your choice of dominant and recessive alleles above, does the appearance of the corn fit with your expectations for the F2 generation? So look at that chart that you just filled out with the phenotypes, remembering, of course, phenotypes are words, and do they match your expectations as you look at the images of the corn that have been posted for you? Now, for each of these, 3 and 4 both go on that chart that you're seeing on this page. You need to come up with the dominant phenotype and the recessive phenotype for both seed color and seed shape. So you'll write those words in the appropriate boxes, and then you need to assign the letter that you're going to be using for each of the versions or alleles. So under allele, remember that is singular. You should be writing in a single letter for the dominant phenotype and a single letter for the recessive phenotype for both seed color and seed shape. When you move on down to number 5, you are going to be using the letters that you put into the chart above, and you'll be using those letters throughout the rest of this experiment. And you want to write out the genotypes of the parents and the F1 generation for both seed color and seed shape. Now, this is a monohybrid cross for both of these, so on each of those lines I should be seeing 2 letters. Next, I've jumped ahead to the very last page, and I want you to take note of table A3 at the bottom of this page. We will be getting back to this in a little bit. But you will see that you have a chart with the 4 different phenotypes that you can have for the F2 generation: dark smooth, dark wrinkled, yellow smooth, and yellow wrinkled. And you have to come up with the number of seeds for each of these 4 different phenotypes. And if you look at number 15, you will see you need to count a minimum of 200 seeds. So when I add up all those seeds and I look at the total, I should see 200 or more on that last row. So how do we do this? Here is one of the images that you will find in the folder on Moodle. You will have to look at several images as you need a total of 200 seeds minimum for this experiment. But I want to talk about what you will see and how you should be counting your seeds. First, it does help to make a chart with the 4 different phenotypic variations, and you can see the blank chart here on my screen. When I look at the corn, what you want to do is be able to identify the 4 different shapes and colors of corn. I put up here and labeled what the wrinkled ones look like. So they look a bit like your molars with dentition and ridges. And then the smooth ones. The smooth will oftentimes have a single divot or line going down the center, sort of like I press my nail into the center of that kernel of corn. Those are still considered smooth. And as you go along here, you will notice a bunch that are dark and a bunch that are yellow. So I can figure out which one the dark smooth, the dark wrinkled, the yellow smooth, and the yellow wrinkled are. Now, I can show you what's going to happen with the row that I have labeled as being counted in this talk. On this image, there's 3 or 4 rows that you can easily count. Once you're finished with those rows, the ones that are in focus, and you can see the entire kernel, you'll then go on to your next image that is found in that folder. And when I look at the row being counted, I can see it starts with a yellow smooth. I then have 2 dark smooth, and then a dark wrinkled. And I would start to make hash marks in my empty table. Now I went down that entire row and I gave you the results from that row. I had 17 dark smooth seeds, 6 dark wrinkled, 9 yellow smooth, and 1 yellow wrinkled. This is a good start, but it's nowhere near 200 seeds. That means I have to keep going to the next row and then the next row and keep on going. I'm now going to jump back to page 2, number 6 at the top. Here you can see I need to write out the genotypes of the F1 generation and the parent generation again. Except this time I'm using both traits, both genes. And so on each of these lines I should have 4 letters. 2 for seed color, 2 for seed shape. I'm then going to continue down and I can start to see that I have Punnett squares to fill out. When I read questions parts 7 and 8 telling me what to do, I will see that this is a monohybrid cross and I'm just using the letters for seed color that I designated in the table on page 1 in the key. On the outside of monohybrid crosses, remember 1 letter, 1 for each gamete, egg or sperm cell, and in the center I should have 2 letters, 1 for each potential zygote or potential offspring. Remember, each of those boxes is a 25% chance, and that's what my prediction is talked about in part 8 on this lab. And I can see I have 2 choices based on table A1, dark seeds or yellow seeds. And so I want to write in the percent based on my Punnett square, percent that should be dark, and the percent that should be yellow based upon my results. Remember, in these Punnett squares, the outside of the square is the F1 generation, that heterozygous generation, whereas inside the squares, inside those boxes, is the F2 generation that you just spent time counting. Okay, and remember that's the mixed generation where you get all variation. You're then going to go to table A1. Now remember, you've already counted your seeds. And so on that table A3, the first two lines say dark smooth and dark wrinkled, but they're both dark. And so I can add those two numbers together to come up with the total number of seeds that are dark. And I can do the same thing for the yellow, which are the last two lines on table A3. I then, as before, come up with the percents. The total number of seeds counted should always be the same throughout this experiment. Now when I come up with the percents, remember it should add up to 100% when I fill in that box at the bottom that says total. However, depending on how you round your percentages, it could be 99 to 101%. If you're outside of that range for the percentages on any of these three tables, something went wrong, and you should go back and double check all your math. Now when you look at the conclusion, the same thing is going to be done for your conclusion in three spots, both for seed color and seed shape, and then for the dihybrid cross. And you can see 11 says what you're supposed to be doing. First off, you're supposed to say, do my results agree with my prediction? And so maybe I said I had 150 dark seeds, and when I divided that by 200, I got 75%. That's what I actually got on table A1 from my counting of the actual corn. But maybe when I go to the Punnett square and my prediction was only 50% dark, well, at that point I would say to part 11A that no, my result did not agree with my prediction. And then I would say, how well did it agree or disagree? And I'd say, well, I predicted I'd only have 50% dark, but 75% of my seeds were dark, a difference of 25%. And then C, can you think of any ways that you can improve how your prediction from your Punnett square matched up with your actual results from you counting those corn seeds? And you need to write it out. Never say, no, there's no other way that I can think that this could be better or that my numbers can match up more or less. Okay, so please don't forget to answer all the parts of the conclusion. This is again another part where people tend to lose points just because they don't answer everything. And I don't want that to happen to you. Now as we go down, we'll see that we're going to do the exact same thing for seed shape, and you're going to repeat the steps 7 through 11 that we just did on the previous page. Only we're doing shape instead of seed color. And we're finally back at that last page again. We've already talked about filling out that table A3 because we got all the information we needed for our other two tables off of this one. But don't forget, you need to fill in the Punnett square under parts number 14. And with the dihybrid cross, remember from the example we had at the beginning of this talk, the outsides should have two letters, one letter corresponding to the seed shape, one letter corresponding to the seed color, and we went through how to come up with those letters when we came up with the gametes. And then inside those boxes, the potential offspring, you should have four letters, two corresponding to seed shape, two corresponding to seed color. And then I have my predictions off to the side. Remember, these should be in percentages. When I gave my demonstration earlier, I did it in fractions. You need to change that into percents. Remember, each box is 1/16th chance of landing there. which means you have a 6.25% chance of landing in any one of those boxes. You need to change it into percents because, again, you're going to be doing your conclusion, the same 3 questions, comparing your prediction from your Punnett square to your actual results from counting the corn kernels from your table. The very last thing at the bottom you'll see is an extra credit. 2 points extra credit, 1 for each question. How do Punnett squares represent meiosis? And how does the process of meiosis explain your results? So question number 1 refers to the Punnett square on this page, and question number 2 refers to table A3. With this extra credit, it is possible to get a total of 27 out of 25 points on this lab. And that is the end of this particular recording.